My topic is actually the about gut microbiota, unraveling the secrets of gut microbiota. To be honest, that you cannot, if you are going to talk about gut microbiota, this 1 hour is not enough. So my outline of my talk is actually I will just describe briefly about this human microbiome and evolution, and also the origin and development of gut microbiota in infants and children, and also the factors affecting gut microbiota development. And early gut microbiota helps maintain health throughout life, and the nutrition shapes the gut microbiota in early life and beyond. So these are the topics that I am going to discuss. Now, about the human microbiome, that is true, we are born 100% human, assuming that we are sterile at birth. But we die 90% microbial because if you take, for example, in our body, we have 10 trillion cells of microbes, but host cells are only 10. So that means 10 times our entire body number of body cells multiplies it to 10 is the number of bacteria in our body. Right, so that you can see. So therefore, when we are dying, we are 90% microbial, only 10% human. And also, it is very surprising that we have, if you take the microbiome, we have 300,000 genes, but we have only 29,000 genes in our body as humans. So genetically also, we are only 10% human. We are 90% bacterial. Right? So that is what really all about our microbiome. This is what it says. So by numbers, if you say you have 10 trillion bacteria, 100 trillion, 10 to 100 trillion microbes, 90% of our body consists of that, right? We have only 22,000 genes, whereas we have 30.3 million genes in our body. So actually, genetically speaking, we are 99.9% bacteria. So actually, human beings we are living in a bacterial world. That is all about human beings. So if the 4 billion years of the earth equal to 24 hours, the man would appear at 23 hours, 59 and 30 seconds. And the Homo sapiens at the last 5 seconds. So that is all about our humanity. So it's just a passing episode in the life of bacteria. So these are all about bacteria. So that is why people now believe we are not Homo sapiens, but we are Homo bacterians. So we are controlled by bacteria. It is all about it. So microbes make man. Now my second part of it is origin and development of gut microbiota in infants and children. Now this is the various stages of gut microbiota development. If you take one by one, colonization at birth. This is very interesting because human beings are normally supposed to deliver vaginally. But now things have changed so that now we have more cesarean section deliveries sometimes than vaginal, but we are meant to be born through the vagina. So the first bacterial colonization of the infant gut typically starts at birth. Well, it is not really so. I'm telling you why. Because the studies had shown that the female, when the baby is born, the placenta also has what we call placental microbiome. So placenta also has bacteria. We thought placenta is sterile. But now we know placenta is not as sterile as we thought. There are placental microbiome. And the beauty of the placental microbiome is that it is not the gut microbe which is there in the placental microbiome. The placental microbiome is actually what the mother is having with the mother's oral microbiome, what she possesses in the mouth. And now, and now we have found that if you have dysbiosis in the oral microbiome, that is, if you have pathogenic bacteria in the oral microbiome, that the placental microbiome also get infected with the pathogenic bacteria, leading to chorea, all the, amnionitis and all that, placentitis, and etc. And now they has found that that is one of the major causes of prematurity, oral dysbiosis, leading to placental microbiome dysbiosis. So that is one of, so therefore that we was, as we say that the baby is born sterile, it is probably not so, because they found even the baby's gut sometimes had bacteria or sometimes fragmented DNA from the mother. So we are truly not sterile at birth, but to be, that is to be very precise. But generally speaking, we are sterile at birth, and the baby get the first colonization starts through the vaginal uh, vagina. So the inoculum is acquired there. Subsequently, microbe continue to acquire from the mother, but also from other family members and the environment. Then, after the baby is born, from the age of 4 to 6 months, the gut microbiome is relatively simple. This is in accordance with the infant diet composed of breast milk. So, the ideal source of nutrition in infancy or infant formula, the adequate substitutes in the absence of breast milk. Bifidobacteria are the key inhabitants of the infant gut, particularly if the baby is breastfed. This is very important. It's the bifidobacteria which is there in a breastfed baby. Lactobacilli are not a dominant taxon in the early life gut, in the first in the newborn period, but late, not the newborn, in the neonate, but later it plays a very important role. Now, this is the point I want to tell you. Now, I said that the earliest, the first inoculum is at birth through the vagina. Then the baby's breast milk, the breast milk we thought is sterile, but breast milk is not sterile. Breast milk, the mother gives about 10 to the power 8 bacteria every day to the baby. Perhaps much more than Mahinda Rajapaksa wealth. Right? And the 40% of babies' gut microbiota, it forms 40% of the microbiota in the gut. And 30% comes from the milk directly, and the rest from the nipple. So it is not the contaminants outside, but it is through the breast milk itself you get bacteria. And the studies have shown there are more than 500 species, and all of them are essentially non-pathogenic. So they are good microbes. Right? Then what happens with the complementary food, that it really started getting changed. Gradually, it started changing. And then you have this, after 28 days, you get the changes. And by about 2 years, you get the adult microbiome in our body. So this is what it is actually. So this is another important point now. My third part of it is, what are the factors affecting gut microbiota development, or how are the things going to get developed? Already I have spoken something, but normally speaking, what are the important things? Maternal factors and mode of vaginal delivery or mode of delivery, whether it is a vaginal delivery or a cesarean section delivery. And also, it is the maternal factor, the maternal diet, the taking antibiotics by the mother, all kinds of things. And if the mother, very often, what happens in Sri Lanka and even other countries, when a lot of mothers do have what you call sort of dyspepsia, so they started giving proton pump inhibitors. That will cause actually gut dysbiosis in the mother, and that will lead to dysbiosis in the baby as well. So maternal factors are very important that shapes the gut microbiome. Then the other factors, of course, the prematurity. Antibiotic use not only during the delivery but also after. Soon after the baby is born, when there's RDs and all that, we start. So prematurity itself changes it, and during, particularly during prematurity, you may not be able to give sufficient quantities of breast milk, and you may tend to give formula fluids or IV fluids. And also on top of that, we give antibiotics. And then the third component I told you about, the breastfeeding. And then after 2 years, the diet what we give. Now, I just want to tell you a very interesting point at this stage is this, the mode of delivery. I told you that if you are a vaginal delivery, you have mainly bifidobacteria colonization. I hope that I have a slide there if I don't miss. No, I think that slide is not there. This is very important point I wanted to tell you, because there was a study done in Texas, and I just forgot the name. It's called Baylor College. Very interesting study. In this Baylor College, what the study that they did was, all the mothers who are supposed to have elective cesarean sections, they have taken a vaginal swab about 1 hour prior to the cesarean section. Just take a vaginal swab, put it through the normal saline. So as the baby is born, as soon as the baby is born, they just have to put that swab over the lips of the baby. Just a simple thing they did. And they found something very interesting, that is that those babies who are born by cesarean section, but their gut microbiota within the first 28 days is exactly like that of a vaginal delivery. And actually, this article is still there, and then the people were asking questions, but this particular lady who did this study says that a day will come that this will be a routine procedure in the theater for cesarean section babies. So the point I want to drive is that even if it is a cesarean section delivery baby, still we could make that happen. So maternal factors I already discussed. I don't have that much time. And this is the cesarean section babies I wanted to tell you. What happened with the cesarean section, that there will be no contact with the vaginal flora. So that it will affect the health, that will affect the gut microbiome, and which we call dysbiosis. You get altered gut microbiome, and that leads to diseases. Whereas if there's a vaginal delivery, you get the normal healthy gut microbiota, and that will protect the baby. Then prematurity. Now, there are a lot actually I can tell. I have no time to discuss about cesarean sections and the incidence of diseases like atopy. No, the studies have shown that cesarean section babies have increased tendency to have asthma, hay fever, and eczema, hay fever, and asthma in comparison to breastfed babies. So there are so many other stuff. Now, prematurity, as I told you, there's high rate of cesarean section, antibiotic use, less breastfeeding, and increased exposure to hospital environment. So you can get all kinds of hospital-acquired infections and immaturity of the gut. So all these things lead to what you call dysbiosis of the gut. Antibiotics, I'm sure that I need to talk a lot about it. I mean, very obvious. And this is what is why it is so important. I will tell you that the gut microbiome in the first 6 months of life is the most essential component of human life. Because that period, that 6 months gut microbiota really shapes us. Who we are today is actually our gut microbiota in the first 6 months of life. If I have time, I will tell you about it. And that is what is very important for you to remember. So you must never ever distort gut microbiota in the first 6 months of life unless it is extremely important and life saving. Unfortunately, what happens today, what happens if a child with 2 months or 3 months have cough and cold, what to do? You give antibiotics. So they go to the doctor, there's no improvement. The parent goes to the second doctor, he goes to the file and says, oh, this antibiotic. So he gives a different antibiotic. And it just goes on. Children with anyway, children breastfed babies tend to have loose motions. So when the parents get panic, they go to the doctor, he gives antibiotics. So we are misusing so much of antibiotics in the first 6 months of life that really disturbs the child's actually overall health, lifelong overall health, which I will tell you in later. So therefore, because you all are budding pediatricians, never ever give antibiotics to a child in the first 6 months of life because they will never ever be able to bring it back, that gut microbiome. Whatever you do, you cannot bring that gut microbiome back. I will tell you why. And therefore, please don't give antibiotics unless it is life saving. This is one of the very important messages I wanted to tell you. Breastfeeding. Breastfeeding has the key role in establishing the infant gut microbiota. Key role. Because it's initially the first 28 days bifidobacteria, predeterminant, then you get lactobacilli and others. And the formula-fed infants harbor more diverse microbiota, but not really even though they are diverse, because it's like if you give breast milk and then complementary feeding, they have more diverse diversity become expanded after 6 months. But what I'm telling you, in the first 6 months of life, in the first few months of life at least, the breast milk gives a perfect combination of microbiota, mainly bifidobacteria. Right. So the complementary foods also subsequently give fiber, and then there will be changing in the colonization. And that diet is very important because we don't expect the baby, as I told you, to have. We need to have a change of this gut microbiome. That is what naturally happens. So the first 28 days breastfeed feeding induced gut micro slowly get changes after 6 months. Till 6 months it is there, but after 6 months slowly changes into adult gut to gut microbiome by 2 years. So that is a very important change. That change should happen. So for that to happen, we should give complementary feeding, also healthy complementary feeding. Particularly, you have to give a lot of fiber and antioxidants. So early gut microbiome helps maintain health throughout life. That we'll discuss what. Now, this is the most important part of my topic. Healthy, resilient gut microbiota provides four important things, actually three important things, to be honest. One is what you call immune tolerance. The other one is called actually it teaches the host or the baby to develop immunity, and also it really helps to prevent entry of pathogenic organisms into the body. But moreover, it also helps to identify and recognize what is mine and what is not mine. And in addition to that, there are a lot of health benefits which I'm going to talk to you. Now we'll just say a little bit about the immunity. Now, there are three components in the immunity. The first component is actually the defense. So the body recognizes what is foreign or what is alien and started producing a host of mechanisms, including chemotaxis and the leukocyte activation, antibody production, and all kinds of immunity. And then histiocyte, the tissue histiocyte also more, modulated into the brought bring into the place. So there are so many things happening with the immune cells during that whenever there is an infection. So the body teaches, the microbiome teaches the gut how to do that. So that is number one. Educating our immune system. Then also it educates, very importantly, how to build tolerance. For example, now say for example, you are having gut microbiome. I told you every 10 day, mother gives 10 to the power 8 amount of bacteria to the baby. And they thrive in the gut. So if the body does not recognize that it is important for me, and body start producing antibodies against them or react against them, we would not have any gut microbiome. They all will be destroyed. So that is tolerance. And also food. We know our food contains all kinds of proteins and other allergens. If those things are getting into the gut, and as soon as the body recognizes that is not mine, start producing antibodies, we will never be able to have, we cannot eat because we will have reactions all the time. That is what atopy means. Every person is not atopic, they can tolerate certain proteins and other stuff. So that is tolerance. So that is being built by gut microbiome, because gut microbiome teaches them, the gut, that this is mine and this is actually very important for you to recognize. So it start teaching to fight against the bacteria, and also teaches to build up tolerance for important things, food and other important bacteria. And thirdly, it teaches you to recognize what is mine and what is not mine. So if you lose that, we start producing antibodies against that. That is called autoimmune diseases. So the proper gut microbiota in the first 6 months of life is so essential to build this perfect immunity, immune tolerance, teaching immunity to fight against bacteria, and also to recognize what is mine and what is not mine. In addition, that they have this antipathogenic, antibacterial activities and other health benefits. If I have time, I will tell you. So this is all what I have told you right now is just discussed there. And also, the gut mite, particularly lactobacillus, not the bifidobacteria, lactobacilli, various types of lactobacilli is now known to protect you from getting infections in the gut. So they themselves act in a different way, various ways they act. And one of the ways thing is what they call decoying, that when a pathogen gets into our body, into the gut, they have what you call glycan receptor, and they get adhered into the glycan receptor of the gut mucosa, and then get entered into the body. That is how they get into the body, pathogenic bacteria. But sometimes they found most of these common cells, particularly lactobacilli group bacteria, they actually block the receptor by already getting adhered into that. So that the pathogenic bacteria can't find a part to glycan receptors to join. All been masked by this common cell bacteria. That is one way. There are various other ways, but this is just one way. Right. So training the immune system, I think I just I don't have time. So this is how it is being run. And this is another important point. How it is being done. It is important that during early life, especially during weaning, immature system is challenged and differ with different stimuli, including pathogens, allergens, and food danger. Now I'm going to tell you something which probably you don't know. Now this pathogenic, now the studies have now shown that the breast milk has maternal DNA, bacterial DNA. And also they have found that even as soon as the baby is born and in the baby's blood has maternal gut microbial DNAs and other particles, DNA particles. So that these things are the ones that really build up this tolerance. Even in utero, and as soon as the baby is born, also all these DNA particles are there. The microbial DNA particles are there, and they will actually help to develop immunity. Now that comes to land tolerance. That comes to a very interesting study which was done by Kaliumaki in Germany. The study called Genie study. You can go through that study. Genie is actually very big project, but this is only part of Genie project. It's a very interesting study that they did. What they did was that they got these people who are allergic to peanut. There's a strong family history of peanut allergy, either a sibling, mother, or father having peanut allergy. And the newborn baby, as soon as the baby is born, and as soon as they pass 4 months of age, they started giving a peanut, small quantity at a time for this child. And then they did a control study where they give peanut after 6 months. Right? And this was very surprising. So those children where there's a strong family history of peanut allergy, if you give peanut between the 4 to 6 months, starting with very small quantity, and they will tolerate peanuts later on. They don't develop allergy. Whereas the group where the peanut was, because of the fear the peanut was introduced later, they develop peanut allergy. So that means that there is a window period to train the immune system. And this is very important. That is why all over the world, WHO, UNICEF, when you take all the societies, pediatric societies, say that we should give exclusive breastfeeding for 6 months, except Espagan. Espagan says that you should give exclusive breastfeeding for 4 months, 6 months. But if there's a strong allergy to a specific product, that product should be introduced between 4 to 6 months. Right. So I think I'll just go to, I have a lot of other stuff to talk about. So microbiota development and trained immune system is linked to multiple benefits. I told you number one is immune system. The 3 aspects of immune system. And also microbiota, which is what you call age-appropriate microbiota. I'm saying again, age-appropriate microbiota. First 28 days is not what you have in the first 6 months, and that is not what you have in the first year of life, not what you have in the second year of life. After second year, that's entirely different. So what is important? The important is that, apart from immunity, it supplement mineral absorption, prevent diarrhea, colics, and other disorders during early life, and also constipation. In other words, it maintains the gut motility, reduce the risk of developing allergies, as I told you, gut-related disorders such as inflammatory bowel disease. Now, a lot of evidence is coming up to say that the gut microbiota is important even though things like autism, diabetes mellitus, obesity, Alzheimer's disease, and various other things. And I'm sure that you know about the inflammatory bowel disease study which has been done, which is called actually the fetal microbiome transplants. The people with severe IBD, very severe IBD and IBS, what they did was they made the gut sterile by giving antibiotics and making sure that it is sterile. And then they transplant, because you cannot give orally now. So they transplant during a colonoscopy. Through the colon, they transplanted normal people's gut microbiome. And more than 80% of these people with IBS, its symptoms completely disappeared. And that is why it is called gut microbial transplant. So that means the dysbiosis is the cause of these problems. Dysbiosis is that you are altering the gut microbiota, which should be what is normal, that you have disturbed by giving all kinds of stuff, which I will discuss what causes that. Right. And also gut microbiota, as I told you, prevent people getting direct infection, pathogenic infections. Right? Nutrition shapes the gut microbiome in early life, and that is what is my topic going to be today. Actually, mainly now this is actually not the real slide, you see. But the important thing about slides this stage is actually you have two components in breast milk. If you want, I can organize one lecture on about breast milk only. One day, if you want. I think I have given that talk about 2-3 years ago. But if you want, I can give that talk about breast milk. Now, the component of breast milk, there are two major components, the micronutrients and macronutrients. Primarily, there are three macronutrients, that is proteins, carbohydrates or sugar, and fats. Lactose, proteins, and fat. But if you know that our fat, if you take 100 ml of breast milk, we have 0.9 grams of proteins, right? 1.4 grams of fat, right? And about 7 grams, 3 grams of fat and 7 grams of carbohydrate, for sugar. And the more importantly, we have zero 0.9 grams of proteins, but we have 1.46 grams, almost double that amount of oligosaccharides as a macronutrient. But we know that oligosaccharide is not a nutrient which we digest. We cannot digest oligosaccharides. No human being can digest oligosaccharides. So the question is, why on earth you're having so much of oligosaccharides in breast milk? Number one. Number two important point is that oligosaccharide remains very high in the first 3 to 4 months of life of breastfeeding, and after that it wanes off. So that means the mother is providing this oligosaccharide for a reason. We will discuss what it is. So this is called actually there are about 160 bioactive components of breast milk. HMO is one, but actually HMO is no longer actually that it's a macronutrient. HMOs we can't digest. There's no nutritive value for us at all. So what does it do? It's provide nutrition for the bacteria what the mother is giving every day in the breast milk. So the beauty is that the breast milk does not only gives the bacteria to the baby, but the so-called healthy bacteria, but also it gives food for that bacteria. Right? We'll discuss a little bit about this HMOs. All right. Host microbe crosstalk during and after breastfeeding is very critical. The breast milk supporting the development of a bifidobacterial dominated microbiota during the first 6 months of life. What makes breast milk unique is the presence of human milk oligosaccharide. Remember, it's called human milk oligosaccharide, which cannot be digested by humans, not infants, but humans. So why we call it human milk oligosaccharides? Because that no other animal produces HMOs like humans do. We produce so much of human milk oligosaccharide, whereas primates produce very little, but no other animal produces significant amount of HMOs. It's unique to human beings. So it expands the gut so much so that the people believe now, scientists believe now, this HMOs is unique to human beings, and that is why the human beings have developed with brain and all that. We have developed brain so much in comparison to other animals is because of HMOs in the breast milk and the gut microbiome. Not only the HMO, the gut microbiome, which I will tell you if I have time. So, but HMOs plays a very important role. And what these are there are primarily three types of HMOs called fucosylated HMOs, silated HMOs, and neutral HMOs. Right? So those are the. I think if I am going to talk to you about human breast milk, I will talk to you about that because I don't have time. But remember that there are three primary types of HMOs. Now, this is not the HMOs that the people are talking about. These reps will come and tell you that we have this sachet with bacteria, the what you call probiotic sachet with HMO, with oligosaccharides as well. Those oligosaccharides are not what we are talking about. They are actually fructooligosaccharides and galactooligosaccharides and inulin. Those are not that important. We are talking about entirely different types of human milk oligosaccharides. They are unique to human beings. And also, there are beauties that there are more than 212 at that, I think little more than that, more than 200 types of human milk oligosaccharide has been isolated, and also that they found that most of these HMOs have different functions, roles to play. For example, that if 2FL, for example, is for to produce bifidobacterium, then maybe 3FL, I'm not saying it exactly the same way, but 3FL may be therefore lactobacilli. And then another HMO like, say, the neutral galactose, then tetrose, that is called LNT, that they are responsible for another bacterial growth. It has another bacteria. So that means, and also different mothers have different kinds of composition of HMOs. So what does it mean? That means the mother decides what kind of bacteria to be there in my baby's body. How much of this bacteria, how much of the other. So mother decides the composition and and not only the composition, but that how much of this one, how much of the other one, how much of the composition of the entire bacterial composition in the first 6 months of life has been decided by the breast milk or by the mother. And that is what shapes the life. That is what that baby is going to be, have everything in that baby's life. Overall health and lifelong health is actually with this 6 months of gut microbiota. I think I don't have that much of time, but this is all about this. Now, this is just a little, I just want to tell you about that. I don't know how to get the cursor now. See, These are the gut microbiota, the so-called oligosaccharide in the breast milk act like a prebiotic, providing food to the baby, to the microbes, gut microbes. Then also it has a very interesting phenomena, what you call the decoy effect. That is this one. Sorry about this, cannot show you. But second, B, you can see the HMOs will really attach to the pathogenic bacteria. So by that will be not allow that to attach into the glycan receptor. When HMO is not there, they attach into the glycan receptor. But here the glycan receptor is there. But HMO has really got the behave like a glycan receptor. That is why it is called it's like it behave like a glycan receptor, and then that will be excreted in the stool, so that pathogenic bacteria cannot get into the body whether through the epithelium cannot get into the epithelium. Similarly, sometimes it happens is some of the HMOs are go and attach to the glycan receptor, so that the pathogenic bacteria has no place to come and attach. And also, they found that the HMOs really improve the gut barrier, the so-called the gut barrier for the permeability for the bacteria to get in. And also, they found that the HMOs really, the gut microbiota and the HMOs really modulate the immune system, so that you have a balance of both Th1 and Th2, reducing atopy and other atopic diseases. And finally, they found that some of the HMOs are get absorbed into the circulation of the newborn baby, and there it actually modulates increase the activity of chemotaxis of the leukocytes. And lastly, not the most important latest development, lot of studies been done, and they found that HMO produces not nutrients to the brain, and also HMA produces what you call neurotransmitters to the brain. Right. Now I am going to talk to you about something different. You have prebiotics, you have probiotics, and you have what you call postbiotics and symbiotics. What are prebiotics? It is very important for you to remember. Prebiotics, by definition, are substrates that are selectively utilized by the host microorganisms, by bacteria, conferring health benefits. So here there are three important points. Prebiotic actually confers health benefit by providing something, may not be nutrition alone, but something for the gut microbiome or host microbiome, not the gut, anywhere else in the body, but they provide something for the microbiome for their well-being, and that causes health benefit. That is the prebiotic. Now, the common what is we have seen outside is actually called galactoligosaccharide, fructooligosaccharides, and inulin and so on. But that is not what I have told you about. I told about something very distinct, that is HMOs. So, what are probiotics? This is very important for you to understand. I have seen many times, including doctors, made this mistake. The probiotic is actually live organisms that, when administered in adequate amounts, confer a health benefit to the host. This is WHO definition up to the point. Every word is important. They are live organisms. If they are dead, that is not a probiotic. And also, when administered in adequate amounts. So if you don't give adequate amounts, you don't have any benefit. Number two. And also administered. What does it mean? That means that is not there in the gut. We are giving outside. Right? And that should confer health benefit to the host. Confer health benefits to the host. So that is a prebiotic, it's a probiotic. So therefore, Gut microbiota is not a probiotic. Probiotic has to be administered outside, right? So the problem about probiotic is the effect of one probiotic strain cannot be generalized to another, and the individual properties of probiotic strain must be evaluated prior to clinical application. I'm telling you this point for a very important reason. Now you see, when this, sorry about that, actually, I have to tell you, reps comes to you and the rep will tell you, you see, doctor, this particular probiotic has only one, this one has only two, this one has only three. We have six probiotics, then we have zinc, and also we have oligosaccharides. To show that that probiotic is much more superior than the others, which is not so. Because we don't know if we have three or four organisms together, how they interact in the gut. Whether you have the beneficial effect of what you expected. And also, all the research about probiotics in the world has been done by one single strain, like Lactobacillus rhamnosus, Lactobacillus reuteri, the Saccharomyces boulardii. So there are various forms of bacteria where they have done research probiotics, but this is a single individual. So one single individual shows some benefit. What is the point of amalgamating all these three or four types and say, I say we have got so much of bacteria? That is not going to work that way. So probiotics, as I told you, probiotics used in early life include several stages of bifidobacteria, lactobacilli, such as reuteri and lactis. Bifidobacterium lactis is supposed to be very interesting actually, because they now found that is one of the microbes which is very helpful in preventing other microbes getting into the gut, the pathogenic microbes getting into the gut. Anyway, no time. So what is symbiotics? Symbiotics is actually a combination of prebiotic and probiotic is called symbiotics. Two together, because it will have a synergistic effect. But unfortunately, when we are using probiotic, we are using our prebiotics actually the oligosaccharides like galacto-oligosaccharide and fructo-oligosaccharide, not the human milk oligosaccharides. But if you give breast milk and the probiotics, then again it becomes symbiotics. Postbiotics. This is a very, very interesting topic which is being now discussed worldwide. What are postbiotics? The postbiotics is a preparation of inanimate microorganisms and or other components that confer health benefit. Very complicated, but what does it mean is the component of a postbiotic may contain intact or part of bacteria, or even the intact bacteria, a fragment of DNA, or byproduct, a metabolite, end product of this. Once they die, the end product. Now that is very important, because we initially thought that the probiotics are only the metabolites induced end product. It is not so. Now the point about that is very important, because about the HMOs. So HMOs, when they are metabolized by the bacteria, they produce what you call SFEs, small chain fatty acids. And that causes the stools to be soft. And also majority of these the SFAs like butyrates and all that are neurotransmitters. And I told you that the HMOs are only exclusively for human beings, and those HMOs have been metabolized by the gut microbiome, and then produces these neurotransmitters. And there are lots and lots of studies are coming up to say that the gut microbiota, in fact, builds the microglia in the brain, synapses in the brain, all kinds of things. I have slides to show you, but if you give me time one day about breast milk, I'll show you how the microglia in the brain, how the synapses in the brain, how all these high functions in the prefrontal cortex is being brought about by the gut microbiota. In other words, now the gut microbiota or the gut is called the second brain of humans. First brain is the brain what you have in the head, and the second brain is actually gut. Not the gut, actually the gut microbiota and the byproducts of that. So that means the gut is constantly talking to the brain, and the brain is constantly talking to the gut. Right? Now, just one word about dysbiosis. Dysbiosis is characterized by a disruption to the microbiome, resulting in an imbalance in the microbiota, changes in their functional composition and metabolic activities, everything. So, because of this imbalance, there is a change in the functional composition, metabolic activities, and shift in their local distribution, causing pathological states. Oral microbiome. If you have dysbiosis, it causes caries, it causes periodontal diseases, it causes gingivitis, and I told you it causes premature delivery. Placental microbiome, premature birth, if there's and also cause chorioamnionitis, villitis, torch infections, and preterm deliveries. Vaginal microbiome, vaginosis, Shirtleram diseases, and yeast infection. Skin microbiome, all kinds of problems in the skin. Gut microbiome, and there are plenty. And I'm just telling you, obesity, metabolic syndrome, diabetes, clostridium difficile infection, colorectal cancer, inflammatory bowel disease, and even psychiatric diseases, and as I told you, Alzheimer's. In addition, allergies, acne, psoriasis, atopic dermatitis, all these been caused by actually not only the gut microbiome, but mainly from the skin dysbiosis of the skin microbiome. Right. Right. I think that probably is enough for today. Right. So my key message is colonization of the infant starts at birth, not really, but to understand, seeded from the maternal gut during the delivery. Gut microbiota develops dynamically in the first few years of life, so ultimately resemble that adult microbiota in late childhood. Breast milk is the provides the optimal nutrition for both the infant and its developing microbiome. It is rich in HMO, supporting the development of healthy gut microbiome. Healthy microbiota development in early life is disrupted by cesarean section, lack of breastfeeding, excessive antibiotic use, and among other factors. Healthy development of early life microbiome. This is very important. Healthy development of early life microbiome is linked to multiple benefits, such as providing gut defense mechanisms, helping good digestion, mineral absorption, supporting the development of immune system, the whole thing, preventing immune and metabolic disorders in late life. Bifidobacteria are the key colonizers in the first 6 months of life in the baby. Gut microbiota gradually diversifies from 6 months onward, stimulated by the nutrition complement food. In addition to HMOs, probiotics, prebiotics are used to promote development of healthy microbiota.